Hi everyone, welcome to your online lecture for the course Altered Physiology 1. My name is Dr. Patricia Riley, and in today's lecture we're covering the second part of our topic Altered Cellular and Tissue Biology. And more specifically, we're going to be looking at the topic cellular accumulation, apoptosis versus necrosis, and aging. Now this slide should very much be a review from normal anatomy, but I'll spend a couple of minutes reviewing with you the main parts of the cell because they will come up throughout this lecture and others. And remember, understanding normal anatomy is very helpful in understanding the abnormal, which we know this course is all about. So here we have a cell, and you can see we have the nucleus that contains the genetic material and really controls all the activities of the cell. The cytoplasm through here contains the cytosolic fluid. It's the area within the cell where most chemical processes take place, and it's controlled by enzymes. We have the cell membrane on the outside that controls the movement of substances into and out of the cell, that phospholipid bilayer. We have the mitochondria. So here's one and another, and we know that this is where energy is going to be produced in the form of ATP. It's the powerhouse of the cell. Another important term is the rough endoplasmic reticulum. So you can see the endoplasmic reticulum all through here, but what makes it rough is the fact that it has all these ribosomes that are present all over it, giving it a rough appearance compared to that of the smooth endoplasmic reticulum that lacks those ribosomes. Ribosomes are found, yes, on the endoplasmic reticulum, but they're also found free-floating. And it's the ribosomes that are important for protein synthesis, and so many things in our body are proteins, and so we need to make sure that this protein factory is working properly and has everything it needs to do so. Otherwise, a lot of problems can arise, and we'll see that throughout this course. Another component is the Golgi apparatus. And this is where the proteins, for example, are going to be packaged and set up and prepared for where they're intended to go, which will obviously vary depending on what that protein's purpose is. So they may keep that protein in the cell, or it may be something that's transported out of the cell. The smooth endoplasmic reticulum is going to synthesize non-proteins because it doesn't have ribosomes to do that. And it'll synthesize things such as lipids and carbohydrates. The smooth endoplasmic reticulum is also an important vesicle that's responsible for storing calcium. So a normal cell should be very balanced in terms of the production of the products and the use of the products. And so one of the things we want to avoid then, by keeping this balance, we want to avoid accumulation of substances in the cell that should not be there in any large amounts. And we'll look at that on the next slide. One of the other things I forgot to mention to you here that is important, there are many other structures that are important, but the lysosomes in the cell, shown here, and they contain digestive enzymes that can help clean up the cell, and whether it's old organelles that aren't working anymore or some sort of pathogen that's entered the cell. So I had mentioned that it's important to have a balance between production and use. Sometimes you can end up with accumulations of material within the cell called cellular accumulations or cellular infiltrations. And this can occur in normal cells, but it can also occur during injured states. The two categories of substances that can produce accumulations, including normal cellular substance, so something normally produced in a cell, can accumulate, and you can get accumulation of abnormal substances. Now we're going to look through some potential reasons why there might be a buildup of substance within the cell. And the first is the pretty obvious one, where there can be just an increased rate of production of normal substances. You're overproducing what your body normally produces. Endogenous just means produced within the body. It's made in your body, and if we produce too much of it, it can accumulate. Some examples of normal cellular substances that can build up include water, proteins, lipids, carbohydrates. If we produce more than we actually need, they accumulate. There can also be a situation where there's insufficient removal of a normal substance. So maybe you produce a product and then you can't properly package it and transport it out of the cell. So it could be a transporting issue. If you can't get it out of the cell and you're still producing, then more ends up in the cell, accumulating. An example that you can make note of here is that there's a condition called fatty liver or steatosis. And this occurs because there's insufficient removal of lipids. And we'll see that a little bit more later. In other cases, maybe some endogenous substance that's produced in your body, whether it's normal or abnormal, is not effectively broken down. So maybe there's the lack of the proper enzyme to break down that product in order to properly dispose of it. In a lot of cases, it's because there's a lack of a vital lysosomal enzyme. And this is called storage disease, and that will come up again later as well. Okay, so lack of a vital lysosomal enzyme. We talked about lysosomes on one of our previous slides. In other cases, there could be an abnormal substance, often the result of a gene mutation that can accumulate because there could be a defect in how that protein folds, or a defect in its transportation, or maybe it's not properly degrading. And because of that gene mutation, it builds up in the cell. And then in other cases, there's harmful exogenous materials that accumulate in the body. So exogenous means coming from the outside in, whereas we spoke about endogenous meaning produced within the body. So these are and occurring in the body. This is when things from the outside are coming in. So it could be that you've inhaled something that ends up entering your cells and building up in them. It could be that you've ingested something, or it could even be from an infection. In any case, whatever the cause is, anything that accumulates abnormally within the cell can be problematic. On this slide, we're going to be focusing specifically on abnormal cellular accumulations. That is, accumulations that are going to have a negative impact on the body when they occur. We're going to go through each of these examples so you can see them labeled 1, 2, 3, and 4. In this first example, this is showing you, for simplicity, a normal cell, but specifically it's showing you a hepatocyte, which is a liver cell. What we can see happening here is all of these yellow markings, it's lipid accumulation or fat. And so as I mentioned earlier, abnormal accumulation of lipids, it could be because it's not being able to be broken down properly and removed. It could be because it's not being transported out of the cell properly. It can lead to a change in the liver, referred to as fatty liver or steatosis, shown here on the left. People with a very poor diet can see changes such as this. People that drink alcohol can see changes such as this, but removal of the inducing stimulus can allow the normal metabolism to resume in many cases. This second example is showing you in this normal cell here that there was a protein mutation. And so now we're dealing with a defect in that protein, in how it folds, in how it functions. And that can also lead to accumulations of that protein within the cell because it's not able to do what it's supposed to do. It's not able to transport and go where it needs to go, so it accumulates. And so it's not just an accumulation of proteins, it's an accumulation of abnormal proteins. Now onto this third example. We know, and this is representing a normal cell, that enzymes are needed to break a complex product into its more small soluble products. And that's part of normal metabolic processes. But what happens if that enzyme is missing? As I mentioned on the previous slide, when there's an enzyme issue, a lot of times it's a vital lysosomal enzyme. And if that's missing, lack of that enzyme, then we have this complex substrate that can't break down into its soluble product. and we end up with accumulations of this endogenous substance within the cell. This is referred to as storage disease. More specifically heard of, you'll hear it as lysosomal storage disease. And that's written here. Okay, so lysosomal storage disease then is an accumulation of endogenous materials due to the missing or lack of a required enzyme to break complex substrates into their soluble products. And in this fourth example, you can see that with this once normal cell, we have an exogenous substance entering the cell. So this would be maybe something that you've ingested or breathed in. So it could be heavy metals, it could be mineral dust, it could even be something relating to an infection, but something from the outside world has entered into our cells and accumulates in the cells because in a lot of cases, it's digestible. We can't break it down. And if we can't break it down, that remains in the cell and accumulates. So that's accumulation of an exogenous material. So this introduced us to some of the mechanisms of cellular accumulations, but let's look a little bit closer now at specific substances and what they can do if they accumulate within the cell. The first one we're going to look at is water. Water can lead to cellular swelling. We know that. That makes sense. If there's too much water in the cell, it's going to swell. And I'll remind you of what we spoke about in class because the concept is important. We talked about hypoxia and how hypoxia is a reduction in oxygen availability to the cell. And we know that the cell, in order to make energy in the form of ATP, needs oxygen to make as much ATP as possible. Needs oxygen and glucose primarily. Now, if there isn't enough oxygen present, then the cell shifts from an aerobic to an anaerobic state. And the anaerobic state is going to produce far less ATP. ATP, remember from last class, is required to operate many things in the body, including sodium-potassium pumps that control the amount of sodium and potassium inside and outside of the cell. So if the pump fails because it doesn't have the fuel to allow it to work, then sodium is going to accumulate within the cell. And according to the principles of osmosis, water will then follow the sodium. And this is going to lead to an accumulation of sodium, but also water within the cell. Remember, also in class, I talked about how when the cell swells, it can lead to the endoplasmic reticulum becoming distended, and then it forms these vacuoles, which enlarge, and then they rupture and form larger vacuoles. And we talked about how with progressive vacuolation, we end up with even more swelling. And this leads to a condition called oncosis, which is really swelling of the cytoplasm of the cell. And this is very dangerous. Now on to the next topic, which is accumulations of lipids and carbohydrates, or the next substance, I should say. There's certain metabolic disorders that can result in the abnormal accumulation of lipids and carbohydrates within the cell. And although these substances can accumulate anywhere in the body, where you see most of the accumulation occurring is in the liver, spleen, and central nervous system. Central nervous system being the brain and spinal cord. And so as you can imagine, if you have accumulation within the CNS of these substances, it can lead to neurologic dysfunction, can lead to severe intellectual disability. So let me give you an example of accumulation of lipids. I'll just highlight that. Accumulation of lipids within the central nervous system. So when lipids accumulate in the central nervous system, we can end up with a condition called Tay-Sachs disease. And this will lead to progressive damage to the central nervous system. People that have this condition, they lack a very specific enzyme that breaks down glycolipids. And because they're lacking this enzyme to break it down, and the lipids can't break down, they accumulate. and cause damage. Now, lipid accumulation in the cell can also lead to excessive vacuolation within the cytoplasm, just like excessive water can. Okay, third topic is glycogen. You should know that glycogen storage is important as a readily available source of energy within the cytoplasm of normal cells. So for example, if our blood glucose levels drop, as it does between eating, glycogen can be broken down to increase blood glucose levels and bring us back to homeostasis. It's a form of negative feedback. Now, similarly, if you've just eaten, excess glucose is taken up by certain body cells and can be stored as glycogen. But when glycogen accumulates within a cell, it can lead to progressive vacuolation, just like we spoke about with water and lipids. And the most common cause of glycogen accumulation is a condition I'm sure you've all heard of called diabetes mellitus. And so this is a disorder of glucose metabolism. Why is diabetes mellitus the most common cause of glycogen accumulation in cells? Well, first of all, glycogen synthesis, which is the creation of glycogen synthesis in the liver is impaired in people with diabetes due to defective activation of an enzyme called glycogen synthase, which produces glycogen. But what we see with people that have long-standing insulin deficiency, that state may actually facilitate synthase activity, producing more glycogen. So in people with long-standing diabetes, or if they're in a long-standing state of insulin deficiency, then it's what's thought is that glycogen synthase is actually being facilitated, producing more glycogen. In addition to this, there appears to be enhanced gluconeogenesis, the formation of new glucose in people with diabetes. So the facilitated action of glycogen synthase, as well as enhanced gluconeogenesis, the production of new glucose, may account for this accumulation of glycogen seen in diabetics. Now, moving on to proteins as a substance that can accumulate and cause damage within the cell. So a few reminders about proteins. They constitute a good amount of a cell's dry weight. They provide cellular structure to cells. We know that proteins are synthesized on ribosomes in the cytoplasm and on the endoplasmic reticulum. We know that proteins are synthesized from amino acids, being the building block of proteins. Now, if proteins accumulate in the cell, it can damage the cell in one of two ways. So we're starting off with accumulation of proteins, and how do they damage the cell? Well, when we break down proteins, so when the cell attempts to, say, digest some proteins, it's going to produce some metabolites, kind of like waste products when they break them down. And these waste products can be damaging. They constitute enzymes that damage cellular organelles. Another way is that excessive amounts of protein in the cytoplasm, they take up room and they push against cellular organelles, disrupting the normal function of other organelles when they accumulate in the cell. So they just take up space, and that can prevent normal communication within the cell. I've discussed lipid accumulation already, but I'm going to elaborate a little bit more on this slide with a few comments that are review. Now, the first part I've talked about a little bit, where metabolic disorders can lead to accumulation. Metabolic disorders specifically caused by inherited disorders where there could be insufficient enzymes or ineffective forms of enzymes. Maybe there's a change in metabolism of the cells within an organ, and we can see abnormal metabolism occurring here, leading to the buildup of lipids within the cell, in this case, the hepatocyte. Now, although lipid accumulation can occur in many different areas of the body, heart, muscle, kidney cells, and so on and so forth, the most common site of intracellular lipid accumulation, or what we can say fatty change, is in liver cells, the hepatocytes. And I mentioned a couple of times already that this is called steatosis. And one of the most common causes of a fatty liver is alcohol abuse. And if a fatty liver is not corrected over time, scarring can develop and it can lead to cirrhosis, which will be talked more about in later lectures. And this is not reversible, but a fatty liver can be reversed. So some examples of the mechanisms of lipids accumulating in the cells. Some of the things that might be seen include just an increase in infiltration of fatty acids into liver cells. There can be failure to convert fatty acids into phospholipids, and this is preferential to convert fatty acids to phospholipids. Phospholipids, remember, are a big part of making up our cell walls. We have that phospholipid bilayer. So instead of converting to phospholipids, they actually the fatty acids convert to triglycerides instead and build up in the cell. There can be a decreased production of apoproteins and lipoproteins in the cell. Apoproteins are lipid accelerator proteins that bind with lipids to form lipoproteins. And then the last point here to be aware of is that if there is a failure in the ability to transport lipids out of the cell, then of course we know they can build up there. Okay, let's speak for a moment about other cellular accumulations. Accumulations of pigment or color can be normal, but they can also be abnormal. Pigments can be exogenous, so they're produced outside of the body, or they can be endogenous, produced inside the body. So looking at an example first of endogenous examples, so produced in the body, this includes melanin, so moles, freckles. Another term for mole is nevi. Melanin will accumulate within epithelial cells called keratinocytes of the skin, and it can also accumulate in the retina. It is an extremely important pigment because it protects the skin against long-term exposure to sunlight, and it's considered really an essential factor in the prevention of skin cancer. Melanin is derived from an amino acid listed here, specifically tyrosine. Another example of pigments are hemoproteins. Hemoproteins are the most essential of normal endogenous pigments. Hemoprotein accumulation in cells are going to be caused by excessive storage of iron, which is transferred to the cells from your bloodstream. Excess iron can build up from really three primary sources: from intestinal mucosal uptake, from tissue stores, as well as overactive macrophages that break up red blood cells and release iron. The last pigment we'll talk about is called bilirubin. Bilirubin is a normal yellow to greenish colored pigment. Excess bilirubin within cells and tissues can cause a condition called jaundice, also and more appropriately called icterus, and that's yellowing of the skin. You'll learn more about that when we get to digestive disorders. In terms of causes, there are many different causes of excess bilirubin, which is called hyperbilirubinemia. Hyper means too much of. Bilirubin we just spoke about, and then emia refers to in the blood. So this is too much bilirubin in the blood. So there are a lot of different causes of hyperbilirubinemia, but for example, it can occur with diseases that affect the body's ability to get rid of it, and obstruction of the common bile duct is another example, but there are others, and again, we will elaborate on that in our digestive disorders lecture. Next up is calcium as a substance that can accumulate in cells. Calcium salts can accumulate in both injured and dead tissues, An important mechanism is by the influx of extracellular calcium into the injured mitochondria. Damage can occur when calcium salts, they start to cluster and then they start to harden, and this can interfere with normal cellular structure and function. Hypercalcemia, so that's an increase in the amount of calcium in the blood that's above normal, can occur in many different diseases and conditions, and an example is aortic valve calcification, which is shown here in this slide. Here you can see some of the calcifications occurring at the level of the aortic valve. The final type of cellular accumulations on this slide is called urate. And in humans, uric acid, or urate, is the major end product of purine catabolism. Purines are natural substances that are found in the body, and when they're broken down, they produce urate. And in normal circumstances, our body will be able to get rid of it through urination or with a bowel movement. Now, sodium urate crystals are the sodium salt of uric acid crystals, but normally sodium urate concentration is considered to be stable in the body, and disturbances in maintaining sodium urate levels can result in hyperuricemia, so high levels of uric acid in the blood. Now, this can lead to the formation of crystals from the sodium urate, and the crystals can land themselves into tissues and cause painful disorders that are collectively referred to as gout. Gout, you can see here, this is a common location of gout in the foot, and you can see that it's actually caused damage to the joints within the big toe. Let's return to calcium for a moment and discuss the effects that it can have when it infiltrates the cell abnormally. And I want you to think of free cytosolic calcium as being a very destructive agent in the cell. Now, calcium ions are critical mediators of cell injury, and they're usually maintained at low concentrations within the cell cytoplasm. But in an injured state, calcium is going to continue to be released from organelles such as the endoplasmic reticulum and the mitochondria. So again, with cellular damage, calcium is going to be released from the mitochondria and the endoplasmic reticulum, and it can infiltrate the cell from outside of the cell as well. So when this calcium is released after cell damage and we have all this free roaming calcium, it has the ability to activate a variety of different enzymes, protein kinases, phospholipases, proteases, and endonucleases, so all of these are enzymes that have damaging effects on the cell, some of which are listed here at the bottom. So the bottom line is that when a cell is injured, the endoplasmic reticulum and the mitochondria are going to release release calcium into the cytosol of the cell. And remember with the mitochondria that in addition to it being a powerhouse, it also helps regulate calcium. And so when it's damaged, calcium's not being regulated. So you get this just constant increase in calcium within the cytosol of the cell. And we know that calcium causes activation of enzymes that lead to further destruction. We have spoken about numerous methods that could lead to cellular death by injuring the cell, but how exactly do these cells die? We've talked about cellular adaptation and cell injury, and that once a cell's injury becomes irreversible, then cells are forced to die. Cell death has historically been classified as apoptosis and necrosis. Starting with apoptosis, it is known as a regulated or programmed cell process that's characterized by the dropping off of cellular fragments called apoptotic bodies. So here we have a normal cell going through the process of apoptosis. And this is showing the fragmentation happening of the cell and the formation of these apoptotic bodies. So too little or too much apoptosis is linked to many disorders. Apoptosis is considered an active process of cellular self-destruction that we call programmed cell death. And it's indicated in both normal and pathological changes. Cells need to die. If they don't die, then we get this endless proliferation that can lead to these gigantic bodies because we're not getting rid of old cells, but we're continuing to produce new ones. The activation of proteases, so these are enzymes, are going to contribute to what we call kind of a suicide cascade, where we're getting cleaving of proteins, where you have stoppage to the life-sustaining activities within the cell, ultimately killing it. An important feature of apoptosis in comparison to necrosis is that it affects single scattered cells as opposed to neighboring cells. So in other words, you're going to have apoptosis of this cell. You're going to have apoptosis of that cell over there, as opposed to apoptosis of a whole bunch of different cells all near each other. So in other words, it's a very nice, neat, particular process. Now moving on to talking about necrosis, a more messy process. Cellular death eventually leads to cellular dissolution or necrosis. Necrosis is defined as the sum of cellular changes that occur after local death and the process of cellular self-digestion. And that is known as autodigestion or autolysis. And those terms will come up on an upcoming slide. So cells actually die long before any necrotic changes are noted by microscopy. And so remember, I said here that it's the sum of cellular changes after local death. Necrosis compared to apoptosis is considered a passive or accidental process of cell death occurring after there's been some sort of severe and sudden injury. So for example, we could have ischemia, which is a reduction in blood supply to a cell, the presentation of toxins, infections, some sort of trauma. The structural signs that indicate that irreversible injury and progression to necrosis are occurring are that you'll see dense clumping and progressive disruption of both the genetic material found within the nucleus, but also disruption of the plasma membrane of the cell and the membranes of the organelles themselves, including that of the mitochondria. And because membrane integrity is lost, necrotic cell contents, so all the necrotic components within the cell can now leak out and can actually cause the signaling of inflammation in the surrounding tissue around it. So it's a very messy process. Now, even though I had mentioned that necrosis is thought to be this passive or accidental cell death, it's interesting to note that there is emerging evidence to show that programmed necrosis might actually be a thing, that maybe it's associated with certain pathological diseases. Maybe it's involved in certain viral infections, as opposed to being what it was thought to be just some random event that occurs. Is it actually programmed in the body to happen for a reason? And you can see when you look at this slide here, it looks just a lot messier on the necrotic side compared to that of apoptosis. I won't be going into the comparison on this slide. This is here as a bit of a summary for you, shown in a bit of a different way. Just note that it's flipped on the slide where necrosis is shown on this side and apoptosis on the other side. But there's also a helpful chart in your textbook that can help you distinguish between the two. But again, you can see necrosis is a much more messy process affecting a group of cells compared to apoptosis, which is a neater process affecting single cells. So you can see neighboring cells are impacted here, whereas they're not with apoptosis. Okay, this slide discusses the process of necrosis. So specifically, let's have a look at what happens to the nucleus. If a cell is plagued with irreversible injury, the cell will die. And in some cells, what we'll see happen is the nucleus will shrink and become this small, dense mass of genetic material. And this is referred to as pyknosis. The nucleus will begin to fragment into smaller particles, sometimes called nuclear dust, and this is a process referred to as karyorrhexis. In the later stages of necrosis, so we're following along later stages here, in the later stages of necrosis, most organelles at this point are disrupted and karyolysis occurs. This process involves dissolution of the nucleus, nuclear dissolution and lysis of chromatin. Remember, chromatin is the material of which the chromosomes of organisms are composed, consisting of proteins, RNA, DNA. Here's the term autolysis I mentioned earlier. I had said that necrosis was the sum of cellular changes that occur after local death, and the process of cellular digestion, which is known as autolysis or autodigestion. And you can see, compared to a normal cell, what's happening here with pyknosis, where you're getting the nucleus shrinking to this small dense mass. You can see some general cell swelling occurring. And then when we look at karyolysis here, you can see you're getting just nuclear dissolution and lysis of the chromatin and rupturing of cell membranes. We're going to look now at the different types of necrosis, and different types of necrosis tend to occur in different organs or tissues, and it can actually sometimes indicate the mechanism or the cause of the cellular injury. So the four types that we'll look at are coagulative, liquefactive, caseous, and fatty. Starting with coagulative necrosis. This primarily occurs in the kidneys, heart, and adrenal glands. Remember, adrenal glands are located above the kidneys. It commonly results from hypoxia due to severe ischemia. So that means that blood flow to an organ or tissue has been severely compromised, meaning that that tissue is not going to get the blood that it needs containing oxygen and other nutrients, and without oxygen it's suffering, or without not enough oxygen it's suffering from hypoxia. Or it can also be caused by chemical injuries such as ingestion of mercuric chloride. Coagulative necrosis is a result of protein denaturation. So what happens is a protein will denature, specifically the protein albumin. Albumin is going to change from its gelatinous transparent state to a firm opaque state. And the area of coagulative necrosis is called an infarct. You probably heard that term before, for example, myocardial infarction. Okay, now I'm going to have a look at liquefactive necrosis. This commonly results from ischemic injury to neurons. So these are the main cells of the central nervous system, and glial cells, which are the supporting cells of the nervous system. And the most common cause of injury is a bacterial infection. So it could be staph, it could be strep, or E. coli. And what happens is there is this autodigestion of the tissue that occurs, and it causes the tissue to soften and liquefy, hence the name liquefactive necrosis. And so the area that's affected is segregated from the healthy tissues, forming cysts. Okay, so the affected tissue is walled off from that healthy tissue, forming cysts. Caseous necrosis usually results from TB or tuberculosis bacteria. So someone that's had a tuberculosis pulmonary infection, which can be caused by Mycobacterium tuberculosis. So this type of necrosis is actually a combination of two types of necrosis, coagulative and liquefactive necroses. And so what happens is the dead cells are going to disintegrate, but the debris is not completely digested. Tissues will actually have this clumped cheese appearance. So caseous necrosis will get this clumped cheese appearance, and then there'll be a granulomatous inflammatory wall that will enclose the area of caseous necrosis. So what happens is the caseous necrosis ends up being walled off. And this can be seen long-term, somebody that's had an old infection, on X-ray of their lungs. Lastly, fatty necrosis is cellular dissolution caused by very powerful enzymes called lipases. So it's an enzyme that breaks down fat. This occurs in the breast, it occurs in the pancreas and some other abdominal structures as well. And what happens is these lipases are going to break down triglycerides, causing the release of fatty acids, because triglycerides are made of fatty acids. And then these fatty acids are going to combine with calcium. They're going to combine with magnesium and sodium ions, and they create almost like a soap. It's called saponification. The necrotic tissue will have this opaque and chalk white look because of that. On this slide, just showing you a couple of examples of what necrosis can look like. Here's the caseous necrosis I spoke about on the previous slide, giving off that yellow, white, cheesy color. Now, this one is gangrene, which this is called gangrenous necrosis, and I didn't speak about this. It's not a distinctive type of cell death, but refers to sort of a larger area of tissue death, and since it's not specific, it wasn't included in the previous 4 types that were discussed. But gangrene necrosis refers to death of tissue resulting from severe hypoxic injury, and it commonly occurs because of conditions such as arteriosclerosis, or maybe the blockage of major arteries to, for example, the extremities, and diabetics are predisposed to this type of necrosis. Okay, we're almost done. I just want to speak for a moment about the effects that aging can have on cell and tissue biology. This diagram shows you the proportion of remaining functions in organs of a person in their late adulthood compared with those of a 20-year-old. So what we can see is a reduction in brain weight. So this is the remaining function. Reduction in cardiac output, reduction in lung capacity. You can see that can drop off quite a lot. And of course, these would be substantially worse in somebody who may have been a smoker their whole life, for example. Reduction in kidney function, kidney mass, I should say, and subsequently kidney function. Conduction along nerves changes. Liver weight reduces. Liver blood flow is cut in half. And there's also a reduction in basal metabolic rate. Now, aging is a natural process compared to disease, which is considered not natural. And of course, lifestyle can really determine if somebody is having just more of the effects of natural aging versus disease. There are some differences that can occur in the rate of aging and in terms of predicted lifespan. Gender can play a role, ethnicity, family history, socioeconomic status, and of course, lifestyle choices. So in terms of what's some of the features that are commonly seen with cellular aging, cellular changes that are characteristic of aging include atrophy, and we know what that means now, decreased overall function and loss of cells, possibly caused by apoptosis, tissue and systemic aging. So progressive stiffness and rigidity is actually common with age. We know muscle atrophy is seen, so that contributes to it, but sarcopenia is seen as well. And this is the loss of muscle mass and strength due to the actual shrinkage of muscle cells. Okay, so sarcopenia is loss of muscle tissue, but it's considered a natural part of the aging process, but it still, nonetheless, has effects. And finally, frailty. Frailty is a common clinical syndrome in older adults, and it leaves a person more vulnerable to things such as falls, functional decline, disability, disease, and even death. But some of the common observations that would be seen would be changes in their mobility, their balance, their muscle strength, their motor activity, cognition, nutrition, endurance, falls, fractures, and a reduction as well in bone density. Okay, on our last slide, going to talk about theories and mechanisms of aging for a moment. Some of the theories involve the accumulation of injurious events, so over time, just wear and tear is contributing to aging, and then it could be a genetically controlled program where we just have a predetermined lifespan. But there are things that also can influence that when we look at the mechanism of aging. So genetic factors, absolutely, but environmental and lifestyle factors are huge as well. So we can make decisions that contribute to our rate of aging and, of course, our incidence of injury and disease. So even though genetics can play a role, we do have it influence ourselves. With age too, there's changes in our cellular control mechanisms, decline in immune function, reduction in the amount of hormones in the body, so things are just not functioning as well as they once did. There's also changes such as degeneration and vascular changes, so joints become more stiff and less flexible, for a variety of reasons, including a reduction in the amount of cushioning and synovial fluid that's present, and a reduction in the ability to properly exchange nutrients and waste. So this is just to get some thoughts going in your head, giving you some ideas on why the body does age the way it does, but rest assured that lifestyle choices make a bigger contributor than ever thought before. So that's the end of today's lecture. Thank you very much for your time listening, and I hope you have a great day.